Okay, chapter 11, meiosis. Okay, we're going to talk about chromosomes during meiosis. We're going to talk about the events that take place, and we're going to explain the difference between meiosis and mitosis. Okay, so meiosis produces gametes, which are your egg and your sperm. And this is what creates genetic diversity. You have half from your mom, half from your dad come together, and you have pairs of chromosomes that create your genetic diversity. This process differs from mitosis in that we're kind of just doing mitosis twice, if you can imagine. You double your chromosomes and then you divide it by 4. Like this. Okay, so you have two, you have a blue and a purple, you double it. Remember, that is S phase of interphase. And then now you're going to go through meiosis 1, which is just like mitosis, right? And you're going to get this. You're going to get one here and one here, but they're doubled, so you're going to do it again until you get one of each. The thing to know here, do you see how we did the crossover? We made this tetrad, they kind of struggled a bit, they pulled each other's little legs, little sections off, and this also increases genetic diversity. Okay? So that is an important part of that process. These are haploid. Right? You are creating haploid cells. The only haploid cells in your body are your eggs and your sperm. Again, mitosis, diploid, meiosis, haploid, eggs and sperm. Again, meiosis. There's an S phase, a prophase, prometaphase. Looks very similar. Metaphase 1, anaphase 1, telophase. This is all just like mitosis, and we just do it again. Okay, meiosis 1, same thing as mitosis. You have G1, where you're just chilling. You have S, where you're doubling your chromosomes. You have G2, where you're adding all the other things that you need to that cell. Okay, so you have, in the S phase, what are called sister chromatids. Okay, again, you have this spindle that holds them together. What is interesting here is you have this tetrad. This is notable about meiosis. And these guys, these little ends here, are going to kind of cross over. And you're going to see, do I have that on the next slide? Yes, look at this. This is crossing over. Okay, so if you look at it genetically with the genes, right? Here's big A, so you got, let's say, this from your mom and this one from your dad. You doubled it. So you have big A, little a, little a. Okay, you're heterozygous for whatever, hair color. Okay, but then during the tetrad crossing over, you get these genes that move around a little bit. So now, remember, you're only going to have one of these. These are going to separate into 4 gametes, 4 sperm cells, if you will. So now this one has big A, big B, little c, right? We've mixed up the genetic diversity a little bit, okay? Recombination. These are called chiasmata, and this is a tetrad of the 4, and that's where your crossing over occurs. Okay. Again, spindle fibers are part of this microtubule, which attach here, and you get this crossing over. Metaphase looks exactly the same. It's just, we're just going to do it twice in meiosis, okay? There's metaphase 1. We're pulling them apart. Here we go. Mostly these are the resulting gametes at the end of anaphase 1. Telophase 1, cytokinesis, just like mitosis. And then, and remember, in plants, we have this cell wall. Let's not forget the cell wall. Here's the animation that we watched in class. That's on Canvas if you'd like to see it again. Now, so we did meiosis 1, just like mitosis, and we're going to do it again. And that's what makes it meiosis, okay? So prophase, prometaphase, everybody line up, right? Now we're lined up, okay? We're lined up with our lunch boxes or our coats or whatever. Now we're separating again, and we're going to get single eggs and sperm, okay? We're just doing the thing twice. prophase, prometaphase. Here it is. Okay, everybody line up. Everybody go to the girls' bathroom, the boys' bathroom, wherever we're going. We're going to separate out. Then we're just doing the whole thing again. Okay, meiosis. Anaphase, telophase, here it all is with the description. Comparing meiosis to mitosis. Both processes divide the nucleus. Mitosis is a single division. Meiosis is twice, right? We're just doing the thing twice. Homologous chromosome pairs form tetrads in meiosis. They do not do this in mitosis. Okay, tetrads only happen in meiosis. In meiosis, crossing over occurs. Doesn't happen typically in mitosis unless something's wrong. Okay, so meiosis purposely sort of makes these sister chromatids that create genetic variation through crossing over. So meiosis reduces the 2N to a 1N. It is a haploid situation. Mitosis doesn't do this. You go from diploid 2 to just another cell of diploid 2. Here's a picture of it. Sexual reproduction. It's important for genetic diversity. Okay? So most animals are diploid dominant, right? Your whole body is diploid, except for your gametes. Okay? So you live all of your life as a diploid dominant organism. Not everybody does it, right? Here's you living as a diploid, and the only things in your body that are haploid are your egg and your sperm. Okay? Haploid dominant, this is a fungi who basically lives like, this is like your sperm got an apartment and lives by itself. Okay? That's what fungi are doing. They do have sexual reproduction, specialized haploid cells, designated positive and negative, joined to form a zygote, which immediately undergoes meiosis to form haploid cells. Okay, so this is what it looks like. This is most of their life here as a haploid, right? This is like your sperm and egg gets its own apartment and lives like this. You do get a zygospore, but then it just goes right back into meiosis. Okay? Alternation of generation. This is plants and algae. I'm going to throw some algae in there. Okay? This is what this looks like. Okay? So here's your diploid stage. Right? Here's your little fern frond. Here's your sporangia that are on the bottom of your fern frond. Okay? You start out as a zygote, make a little fern frond, sporangia. This is your diploid life. But then these sporangia throw out some spores. Those spores go through mitosis and make these little gametophytes and sporophytes. And they live all on their own, right? Your egg and your sperm got an apartment and they're just chilling by themselves. They're doing their own thing. They probably have jobs. I don't know. Right? So here is your egg and sperm just chilling, right? They got their own place. They're in college. I don't know what they're doing with their lives. Okay? The sperm and the egg, they eventually fertilize and the process starts over. Okay? So half their life they're diploid, half their life they're haploid. Non-disjunction. Remember, this is the failure of the homologous chromosomes to separate properly. Okay, normally it should look like this, right? But very often you will get 3 where there's supposed to be 2, or you will get 1 where there's supposed to be 2. This can happen in meiosis 1 and in meiosis 2. So autosomal aneuploidy, these sometimes lethal. You can get trisomy 21, which is Down syndrome. You have Edwards syndrome or Patou syndrome. This is a chromosomal issue. It is not genetic. This is not big A, little a. You can't do a Punnett square with these. Okay, sex chromosomes. This is your Turner syndrome. You only have 1 X chromosome. Or your Klinefelters. You have 2 X's and a Y. Okay, and that is the end of meiosis.